Introduction to the silent threat, hepatitis D. Hepatitis D, often overlooked but potentially devastating, presents a unique challenge in the world of viral infections. Despite its relatively low profile, the impact of this virus on global health cannot be underestimated. This intriguing virus, also known as the hepatitis delta virus or HDV, is a defective RNA virus. Its unique nature sets it apart from other hepatitis viruses, making it a subject of intense scientific interest. This means it cannot replicate on its own. Unlike other viruses that can independently invade and multiply within host cells, HDV requires assistance. Instead, it relies on the presence of another virus, the hepatitis B virus or HBV, to complete its life cycle. This symbiotic relationship is both fascinating and perilous. This dependence on HBV makes HDV a fascinating case study in viral pathogenesis and a serious threat to human health. The interplay between these two viruses creates a complex dynamic that challenges researchers and healthcare providers alike. Imagine a silent intruder lurking in the shadows, unable to penetrate a fortress on its own. This intruder is cunning and patient, waiting for the right moment to strike. It needs an accomplice, another more successful invader, to open the gates and offer safe passage. This accomplice is the hepatitis B virus, which unwittingly aids HDV in its destructive mission. This is the story of HDV, a viral opportunist that hijacks the machinery of HBV to wreak havoc on the liver, the very organ responsible for detoxifying the body. The liver, a vital organ, becomes the battleground for this viral onslaught. The implications of this dependence are far-reaching, influencing not only the virus's transmission, but also the course of the disease and its management. The dual infection complicates treatment protocols and requires a nuanced approach. People with HDV infection always have an underlying HBV infection, adding another layer of complexity to diagnosis and treatment. This dual infection scenario makes it imperative for healthcare providers to be vigilant and thorough in their approach. This interplay between two viruses, one a master manipulator and the other a willing accomplice, creates a formidable foe that demands our attention and understanding. The collaboration between HBV and HDV is a testament to the complexity of viral interactions. The presence of HDV complicates the already serious consequences of HBV infection. The combined effect of these viruses accelerates the progression of liver disease. It can accelerate liver damage, increasing the risk of complications like cirrhosis and liver cancer. The stakes are high, and the need for effective treatment strategies is urgent. Understanding the intricacies of HDV, from its structure and transmission to its interactions with the human body, is crucial in our fight against viral hepatitis and the global burden of liver disease. Continued research and collaboration are essential in developing effective treatments and ultimately finding a cure.
Decoding the enemy. Structure and genetics of HDV. Unveiling the mysteries of HDV begins with understanding its basic structure and genetic blueprint. This journey into the microscopic world reveals the intricacies of a virus that, despite its simplicity, poses a significant threat to human health. This virus, unlike most others, possesses a unique and surprisingly simple architecture. Its minimalistic design is both fascinating and deceptive, hiding its true potential for harm. Picture a tiny sphere, barely visible under a standard microscope, its surface studded with proteins. These proteins are not just structural components. They are the virus's tools for survival and replication. This is the HDV virion, a minuscule package of genetic information encased within a protective protein coat. This coat is crucial for the virus's ability to withstand the hostile environment outside a host cell. The HDV virion's simplicity is striking, its genome consisting of a single-stranded RNA molecule. This RNA is not just a passive carrier of genetic information, it is an active participant in the virus's life cycle. This RNA strand, remarkably small for a virus, carries the genetic instructions for only two viral proteins, the small and large hepatitis delta antigens, or HDV. These proteins are the key players in the virus's strategy to hijack the host cell. These proteins, essential for the virus's survival and propagation, play pivotal roles in hijacking the host cell's machinery. The small HDV is particularly adept at evading the host's immune defenses, allowing the virus to persist and replicate. The small HDV, a master of disguise, helps the virus evade the host's immune system, ensuring that the virus can continue its life cycle without being detected and destroyed by the host's defenses. While the large HDV acts as a molecular architect, orchestrating the assembly of new viral particles. This protein ensures that new virions are correctly assembled and ready to infect new cells. The efficiency with which these two proteins carry out their tasks is a testament to the elegance and cunning of viral evolution. Over time, HDV has honed its mechanisms to become a highly efficient pathogen. Despite its structural simplicity, HDV's genetic makeup holds the key to its dependence on HBV. This relationship is a fascinating example of viral interdependence and co-evolution. The virus lacks the genes necessary to produce its own envelope proteins, the molecular keys that allow viruses to enter and infect host cells. This deficiency is not a weakness, but a strategic adaptation. This deficiency forces HDV to rely on HBV, borrowing its surface antigen, HBsAg, to cloak itself and gain entry into liver cells. This clever strategy allows HDV to exploit the resources of HBV, ensuring its own survival and propagation within the host.
Pathways of invasion, HDV transmission. HDV, much like its accomplice HBV, relies on direct contact with infected bodily fluids to spread and infect new hosts. This means that any situation where blood or other bodily fluids are exchanged can potentially lead to transmission. This stealthy virus, however, faces a unique challenge: its absolute dependence on HBV for replication. Without HBV, HDV cannot replicate or cause infection, making it a satellite virus. This dependence restricts HDV transmission, making it contingent upon the presence of an active HBV infection. Essentially, HDV needs HBV to create the necessary environment for its survival and replication. Imagine a lock-and-key mechanism where HDV represents the key and HBV the lock. Without the lock, the key is useless, and without HBV, HDV cannot unlock the door to infection. Only in the presence of the lock, an individual already infected with HBV, can HDV unlock the door to infection. This symbiotic relationship means that HDV is often found in individuals who are already battling HBV. This reliance on HBV significantly narrows the pathways of HDV transmission, limiting it to scenarios where there is a high risk of exposure to infected blood or other bodily fluids. This includes medical settings where strict protocols are necessary to prevent cross-contamination. The most common routes of transmission mirror those of HBV, involving direct contact with infected blood, primarily through sharing needles or syringes contaminated with the virus. This is a significant concern in communities where intravenous drug use is prevalent. This mode of transmission is particularly prevalent among people who inject drugs, highlighting the importance of harm reduction strategies in controlling the spread of both HBV and HDV. Programs that provide clean needles and educate about safe practices are crucial in these settings. Sexual contact, especially unprotected sex with an infected individual, also poses a significant risk of HDV transmission. The virus can be transmitted through semen and vaginal fluids, making safe sex practices essential. The virus can be present in various bodily fluids, including semen and vaginal fluids, making intimate contact a potential route of infection. This underscores the importance of regular testing and open communication between sexual partners. Additionally, vertical transmission from an infected mother to her baby during childbirth is another possibility, emphasizing the importance of HBV vaccination for newborns. Vaccination can provide a crucial line of defense against both HBV and HDV, protecting the next generation from these infections.
A tale of two infections, co-infection versus superinfection. Understanding the dynamics of HDV infection requires distinguishing between two distinct scenarios, co-infection and superinfection. These terms, though seemingly interchangeable, represent two very different paths of infection, each with its own implications for disease progression and management. Co-infection, as the name suggests, occurs when an individual becomes infected with both HDV and HBV simultaneously. Imagine two invaders arriving at the gates of a fortress at the same time, each contributing to the siege. In this scenario, the immune system faces a dual assault, battling both viruses concurrently. Superinfection, on the other hand, paints a different picture. Here, an individual already chronically infected with HBV encounters HDV. Picture a fortress, already weakened by a prolonged siege, now facing a fresh wave of attackers. This scenario often leads to more severe liver disease, as the pre-existing HBV infection creates a fertile ground for HDV to thrive. The clinical course and outcomes of HDV infection differ significantly depending on whether it's a co-infection or a superinfection. Co-infection, while still serious, tends to be less aggressive, with a higher chance of the body clearing both viruses. Superinfection, however, often leads to more severe liver damage, increasing the risk of complications like cirrhosis and liver cancer.